All right, welcome everyone to another episode of The Abstract, the first one of 2026. Happy New Year to everyone. I'm your host, Shahin Zagros. The Abstract is your trusted podcast for all things brain research. And what we like to do here is to break down recently published neuroscientific papers into four components: objective, methods, results, and significance. My guest today is none other than Dr. Mike X. Cohen, an extraordinary individual with a truly unique career path. Um, I like to think of him as the renegade of neuroscience, and I mean that in the absolute best way possible. Now, before becoming the founder of the very popular online educational platform Sync Express and a best-selling instructor at Udemy, he was just an associate professor at the prestigious Donder Center for Neuroscience in the Netherlands. And then one day from the ivory tower, he decided, that's not for me. Let's pack it up. And today I will gladly drag him back into the world of academia, at least temporarily. Dr. Cohen is also something of a time traveler. It's currently January 8th here in Canada, but where he's joining us from, it's January 9. So, without further ado, let's welcome him back to the past. Mike, are there any winning lottery ticket numbers that I should be aware of?
No, but I think I was happy to welcome in 2026 before you and lots of other colleagues and friends around the world. But thanks for
Wonderful.
inviting me. It's nice to be on here.
It's a pleasure, Mike. So before we get technical with the neurosciencey stuff, why don't you tell us a little bit about who you are, what got you interested in the brain, how you became a neuroscience, and then maybe briefly discuss what kind of inspired you to move to other things.
Whew, yeah, I do feel like it's a very long, complicated answer to a simple question. But briefly, I mean, how did I get into neuroscience? I kind of just fell into it one step to the next. A lot of things in life just keep going by inertia, right? Like the change from any one day is really small relative to the change from the previous day, right? So there's so many paths that we take in life and so many decisions that we make that seem very tiny at the time and just kind of natural and easy. But then after a decade, you look back and you're like, man, how did I get here from there? So, I mean, it's not like when I was a kid, I never had a dream of being a professor that was, or a scientist. That was not a thing in my, sort of mental workspace when I was younger. I liked being in school. I liked being at university. I liked learning new things. I liked studying. I liked writing. I liked coding. And the reason why I went to get a PhD was because I didn't know what else to do. Like, I was thinking, like, after my bachelor's, I was like, man, what should I do with my life? And I like being in school, so I thought, well, I'll go to grad school because what else am I going to do? So yeah, just like one thing led to the next, and then before I knew it, I was a professor. And why, I started in psychology, and psychology is one of these fields that, it's like a default field. If you don't know what you want to study, you study psychology, right? So that's how I got into psychology. And then neuroscience, I got into partly just because the brain is super interesting. Everyone finds the brain really interesting. And also partly because I actually started my PhD studying personality psychology. And I liked the kinds of questions that are asked in personality psychology, but I didn't. I was unsatisfied with the data. I wanted more data. I wanted more detail in the data. I wanted like I wanted more headaches from analyzing data. So that kind of got me into cognitive neuroscience and then systems neuroscience.
Yeah, yeah. Talking about these tiny little changes over time, it kind of reminds me of like integration in mathematics. It's like you have instantaneous tiny little things, and then you just like integrate across the whole thing, and you get like this big picture that emerges.
Yeah.
Um,
Exactly. Yeah, there is really a calculus to life, right? That like very, very tiny changes can end up being just these sort of wild nonlinear functions.
yeah. Um, Mike, back in September, you sent me a private message that really stood out to me. And at the time, I didn't know who you were, but I found your message very encouraging and validating. And if it's okay with you, I'd like to share it with the viewers and the listeners. All right. So you said, "Hey, bro, there's nearly nothing on Substack about neuroscience, actual neuroscience, not pseudoscience with the word neuro in it. Keep up the good work." And then you gave me the thumbs up. Um, like as you pointed out, there are lots of pages and accounts, not just on Substack, but also other social media that purport to be something that they fundamentally are not. Right now, I'm in the midst of drafting an article on this very topic about how you have a kind of academically credentialed individuals who really ought to know better, trying to sell their subscribers and followers what, in my eyes, boils down to the neuroscientific equivalence of snake oil. Um, so before we talk about your paper, like I wanted to ask you, what do you make of these online, I want to say charlatans? I don't know if I'm being too harsh. I don't know if I'm not being harsh enough, but what's your take? Are they just trying to make a living by feeding their followers these innocuous, half-baked ideas, or do you think maybe there's something more nefarious happening?
Yeah, I think probably both. So first of all, there's nothing new about, sort of snake oil peddling in the day of the internet, right? So, I mean, that's been around forever. I guess what the internet and social media allows people to do is kind of supercharge that idea, that approach, and amplify it to a global audience. I think it sort of depends. There's also all this stuff about. So I think my answer to your question, I think there's both. There's people who are peddling or spreading misinformation who have good intentions. They just haven't taken the time themselves to understand the science or to get it directly from actual authority figures. And those are well-intentioned people. And then there's people who are just trying to, make money or get internet fame or something like that by just spewing whatever bullshit comes into their mind. With the former, you hear a lot about like dopamine and dopamine detox and dopamine breaks and things like that. And it's like when I read posts like that and see YouTube videos like that, I think a lot of their advice is great. Like put down your phone, no, don't bring your phone into your bedroom, right? Stop using social media after whatever 9 p.m. Like this is all fantastic. Like take a day off per week, no computers, and go outside and touch grass and play with your dog or whatever. That's fantastic advice. This idea that it's actually like reducing dopamine in the brain, right? It's like complete bullshit. And if it actually did, like if these techniques actually reduced dopamine the way that they say it would, that would be awful. You'd have Parkinsonian symptoms. You'd have like cognitive problems. You'd have depression. You'd have like you wouldn't be able to move and pick up a cup of coffee. Right? So the science that they're claiming is completely wrong, but the advice they're giving is good, right? So these kinds of yeah, I have different. Sometimes I think if it helps someone improve their mental health, then it's okay, right? And sometimes I think, man, I really wish these people would just talk to an actual neuroscientist, right, and get some real scientifically grounded advice before peddling it.
Yeah.
Yeah, I don't know. It's tough to say. Yeah.
Yeah. Yeah, I think a lot of it sometimes, like you pointed out, also boils down to just common sense. Like you said, yeah, okay, put your phone away before you sleep. It'll improve your sleep hygiene. Pardon my French, no shit. I've always maintained, common sense is a free cognitive commodity, and you should always be very skeptical of individuals who are trying to sell it to you. But yeah, I think you and I kind of see eye to eye on this matter. Okay, let's shift gears. So the paper we'll be diving into today is titled "Multiple Midfrontal Thetas Revealed by Source Separation of Simultaneous MEG and EEG," and this work was published in the Journal of Neuroscience back in 2020. We'll have the link available. So let's start with some background information, Mike. Tell us in broad strokes what this paper was about, what the main objective was, the driving hypothesis, and why was there this focus on theta bands, specifically midfrontal theta bands?
Yeah, great. So, there's if you look at the sort of development of our understanding of nature, of the world, there's always this progression from thinking that a phenomenon is like one monolithic thing to appreciating the complexity of it, right? So, like a long time ago, people looked at the sun and they thought that's a deity, and he wakes up in the morning and he goes across the sky and then he goes to sleep at night. And now we know better, right? We know the sun is like this massively complex physical system, right? But if you just look at it like this, and don't look at it too long directly, but if you just look at the sun, it looks like it's one thing. But if you have better measurements and a better understanding of physics, you can appreciate the enormous amount of complexities that go into the sun. And you have that also with like matter, like this is, mass. People used to think mass is one solid thing. And now we know better. We know that mass is like this really it's mostly empty space. It's molecules and atoms held together by physical forces and so on. Okay, so also in neuroscience, right? People used to think the brain was one thing. There's like the pineal gland that's sort of controlling everything. And now we know a lot better. The brain has neurons, right? But it doesn't just have neurons. It has like two broad categories of neurons, excitatory and inhibitory. But even that classification is like way oversimplified. There's dozens of different types of interneurons, for example. Okay, so that, and then, think about like memory, right? There isn't just the memory. There's all these different systems, different kinds of memory. Okay, so now let's get to my little pocket of neuroscience, which is called response conflict and response conflict monitoring. So there, the phenomenon, the behavioral cognitive phenomenon, is that you want to engage a particular action, but instead your body does something other than what you want it to do, right? So tons of examples of this from everyday experience, but one is like keyboard typing, right? So there's like tens of millions of people that type on keyboards every single day. You're writing about like ducks, you're writing someone about how you don't have any ducks to give them. And your finger is, you want your finger to go to the D key, but it actually then accidentally goes a little bit too far to the right, and you're going towards the F key, right? So there is a mistake in your action, and you get immediate feedback. You press F and you see on the screen that there's an F instead of a D. So that is an error, and your brain can super fast, very quickly identify that there is this conflict. This is where the word conflict comes from in response conflict. There's a conflict between the intended action, which was to press D, and the actual outcome, which was that the F was pressed. Okay, so you can have these error signals happening after the fact, after the error was made. But it turns out that there is a system in your brain that is continuously monitoring your actions as they are being sent to the spinal cord, and comparing the outgoing motor plan to the cognitive intention, to the higher level intention. So that system can actually recognize that there is this conflict between the intended action, which is pressing D, and the actual outcome, which is that your finger is moving towards F before you actually get to the F, right? So your brain can recognize that you are about to make a mistake, and then it can very quickly, in like 150 to 180 milliseconds, basically adapt the ongoing action to correct your behavior so that your finger is moving towards F, but at the last possible minute, it gets pushed back. Your brain sort of realizes you're about to make a mistake and it pulls it over to the left. And then you can write about ducks and all the ducks you don't have to give. So how does that process work and what does it look like? So yeah, that is called response conflict monitoring. And by the way, it can happen consciously when there's a big error you're about to make. You are consciously aware that you're about to make an error. But a lot of this is also preconscious or unconscious. So we can measure this in the lab and we can show that you have this ability to recognize that you're about to make a mistake and then correct your actions without being consciously aware that this is actually happening. Okay, so what does this process look like? We call it, when we measure it electrically by measuring electrical activity of the brain with sensors that we put on the head, we call this phenomenon midfrontal theta. And why do we call it midfrontal theta? It's called midfrontal because we measure it from here. It's maximal around here. And it comes from somewhere around the pre-SMA. So it's like if you would stick your finger into your skull, basically, you push your finger into your brain. Don't try this at home, but if you did, then basically where your finger would land, that's basically the source of this signal. And I don't know if this is like just cosmic coincidence, but what do you do when you fuck up, when you make a mistake? You're like, oh shit, like you hit yourself in the head exactly where we see midfrontal theta coming from. So make of that what you will. Okay, so that's why we call it midfrontal. Why do we call it theta? Theta is a Greek letter in electrophysiology, so the study of the electrical dynamics of the brain. You find that electrical activity is very strongly rhythmic. It's oscillating up and down. So the electrical rhythms are going up and down. This reflects coordinated hyperpolarization and depolarization of large populations of neurons. So like thousands to hundreds of thousands of neurons that are firing synchronously, or at least are sort of synchronously active. And their combined electrical activities oscillate like this. And so in the brain, you find oscillations at many different speeds. There are, during deep sleep, you have very slow oscillations. And there are like sensory related oscillations that are much faster. And then the speed of these oscillations is not arbitrary. They're grouped into bins, into relatively small frequency ranges. And there are multiple of them. So the cognitively relevant ranges are given labels of Greek letters just for convenience. So we have delta, theta, alpha, beta, gamma. Those are the common ones. You can think of these as like a little bit like radio stations that can broadcast information in different channels simultaneously. So having activity at different frequencies is a way for the brain to process and transmit information simultaneously in space, simultaneously in time, but then segregated according to the frequency. That's part of it. And also part of it is that the speed of the rhythm is determined by and also in turn constrains the speed of the cognitive process. So if you have a rhythm that's oscillating at 100 milliseconds, then the cognitive or perceptual operations that are happening in that rhythm are also constrained to operate within these windows of 100 milliseconds. Okay, so that was a lot of neuroscience EPHIS background, but basically we see that when you are experiencing response conflict or a full-blown error, the by far dominant, really the only thing that we measure when we control for all these other perceptual motor factors, the only thing we see at the level of the scalp is midfrontal, and it's in theta. So it's around 6 hertz or 6 cycles per second. That corresponds to around 150 milliseconds. If you want to know what that feels like, it's basically the fastest you can clap. So if you think about like how fast you can clap, then that's basically theta. And that is the speed with which this system in the medial prefrontal cortex is sort of clocking a snapshot of what is the action that you want to take and what is the action that is actually sent out to the spinal cord into your body. And when those two misalign, those two signals misalign, that is response conflict, and then you get this big burst of theta activity.
Yeah. Uh, Fantastic starting analogy and explanation, Mike. So in this study, so simultaneous MEG or magnetoencephalography and EEG electroencephalography data were recorded in 9 healthy young adults, and they were performing this conflict-inducing assessment protocol known as the Simon task.
Yeah.
Could you just briefly describe what MEG and EEG are, what brain property each one measures, and then talk a little bit about what the Simon task is and why it was administered?
Sure, yeah. So I can start with the Simon task first,
Yeah, sure.
because it just so earlier I was talking about like pressing keys on the keyboard. That is a great experimental paradigm because it's very real world relevant. Everyone does it all the time. But it also, typing has a lot of like linguistic overhead. So experimentally, what we would like to have is a very simple, very pure, and very fast paradigm, experimental paradigm, where we can really measure this response conflict process in a sort of pure way and be able to control for other confounds. So what we do is there's a collection of tasks that you can use to measure response conflict. I like the Simon task because it's just really great. It just gives a really good response. So here's how it works. The task is super simple. The research participant is sitting in front of the computer screen with a mouse on their hand or a response keypad, something like that. And all you have to do is, if there's a red circle on the screen, you press the left button. And if there's a blue circle on the screen, then you press the right button. Super simple. Couldn't get any dumber than that, right? So one color maps to left, one color maps to right. Now, here's the thing. We don't present this stimulus in the center of the screen. We present it off to the left or to the right. When we have the red circle on the left, then that is congruent because you press the left button for red, right? But when the red circle is presented on the right side of the screen, then you have to press the left button. Now, if we give people a lot of time, we say, take 10 seconds to do this, then there's no response conflict, right? But when we tell people, you respond as quickly as you possibly can, that generates a ton of response conflict because we have this automatic, like collicular, very like low-level mapping between spatial location and response location. When you see something to your right, your entire system orients to the right because you want to see what's going on over there. But then you have to press the left button. So that is what generates this response conflict. Okay, so that's the idea. So we have colors on the left or the right, and they have to press the left or right button. Slightly more complicated than what I explained because we use multiple colors and multiple shapes just for avoiding other compounds. Okay, so that's the Simon task. So now I'll talk briefly about EEG and MEG. When there is, so neurons communicate with each other through electrochemical signaling. And for electrophysiology, what's relevant is the electrical part of that signaling. There is a tiny little jolt of electricity that goes down the neuron axis. So it's like a little bit of electricity going through a wire. And when you have electricity going through a wire, there's a little electrical field that's generated around that wire, and there's simultaneously a magnetic field that is perpendicular to the electrical field. Now, the electrical potential from one neuron is very, very weak. You can measure it, but you need to be inside the brain. You need to have a tiny electrode, and it needs to be really, really close to the neuron. So if you want to measure non-invasively in humans, you cannot do that, right? But it turns out that lots of neurons in the cortex are geometrically aligned, so all of their axons are aligned in parallel. And when they are simultaneously active, all of their individual potentials some and get bigger and bigger, more and more powerful. And then we can measure them from outside the brain, outside the skull, outside the head using electrodes that we put on the skin. So EEG is measuring the electrical signals. MEG is measuring the magnetic fields that are simultaneously generated. There are some subtle differences between what EEG and MEG is sensitive to, but it's not so relevant. What's relevant for here is that these are just different points of measurement that we can get from the brain. And we wanted to measure as many points on the head from the brain as we possibly could. So with EEG, there's high density EEG going up into the hundreds, but typical EEG is somewhere like 50 to 60 electrodes on the head. And MEG is, because it's a slightly different technology, typically measured somewhere, 2 to 300. So we combined these to get as much information, as rich a data set as we possibly could. And we ended up getting somewhere around 330 sensors simultaneously measured from the brain, recorded at, I think it was 1200 hertz. So basically, every millisecond, we take a snapshot of the electromagnetic signals that are happening in the cortex that we can measure from outside the head. So super rich data. And why did we do that? Why not just measure 64 channel EEG like, hundreds of other labs around the world? So now I want to tie back a little bit to how I started this narrative with thinking, this intellectual progression from thinking that some phenomenon is a unitary thing to appreciating that it's a complex set of interwoven pieces. Historically, people have thought that response conflict was one thing. There's one box in the brain. There's one circuit in the brain that does the entirety of this conflict process. And why did people think that? People thought that because scientists are simultaneously very smart and very stupid, right? So we find very simple ideas very tantalizing. So to say that this cognitive process is one unitary thing, and in the brain it's handled by one dedicated circuit, that's just very appealing to us as scientists. And I also used to buy into this, right? So I also used to believe that it really was this one dedicated, tiny, spatially restricted, localized circuit in the medial prefrontal cortex that handles this. And part of the reason why I thought that it was one dedicated circuit and just one unitary phenomenon is because that's what other people were saying. That was sort of just general wisdom in the field. And also part of it is because that's what I observed in my data. When I looked at my relatively low resolution data using relatively coarse analysis techniques, or at least spatial analysis techniques, it looks like it's just one thing, right? And over the years, we started questioning this and started being concerned that the reason why we think it's just one unitary phenomenon is because we're not measuring it well enough with enough spatial resolution, and we're not doing the right analyses to identify the possibility of having more than one component. So that's why we measured the shit out of the brain. We measured as many electrodes as we possibly could with two different techniques, so electrical recordings and magnetic recordings, because we wanted to have as much sensitivity as we possibly could. And it was that set, the combined high density measurements in combination with these sophisticated machine learning multivariate decomposition techniques, that is what allowed us to come to the conclusion that actually we were wrong. And I think the whole field was wrong, that it's not one thing. It's response conflict is actually a really complicated interwoven set of circuits and sub-processes. And if you don't measure it carefully enough, like when you look at the sun with your, squinting your eyes, it looks like it's one thing, but you need better measurements, you need better theory, and you need better analysis techniques to really identify that it is actually a lot of complicated, interwoven, but statistically independent things. I think I may have veered slightly off from your question, but that gets to, I think that helps explain why we did these measurements and also why we needed these, sort of overly gratuitously complicated statistical techniques and so many statistical controls.
Yeah, I mean, the principle of parsimony, otherwise known as Occam's razor, is a great guiding principle when it comes to these things. But like you said, like it's not the end all be all of research. You need to be able to squint a little bit harder sometimes to see layers of realities that are just beyond the surface. It's interesting because like you're describing the Simon task, and to me it sounds a lot like the Stroop task, where you have you'll present the letter red, for instance, but it's written in green font, and then you're either asked to say what color it is or what the word is, and there's like conflict processing that also happens in the brain.
Exactly. Yeah, the Stroop task is a fantastic demonstration of exactly the same phenomenon of response conflict. In fact, so I don't give talks anymore because I left academia. But when I used to give talks, I didn't introduce the Simon task. I introduced the Stroop task. It's so compelling, and you just put the words on the screen. You write the word purple in green font, and I just say like, read that word out loud, and people get it. So I wouldn't say the Stroop task is problematic per se, but it is language based. So that brings on this whole extra dimension of
Right.
complexity. And then there's also an additional risk of individual differences, for example, in like reading capabilities, right? And language. So if English is your third language and you learned it as an adult, you don't have that much of a Stroop task because it's just much easier for you to suppress reading the word and say the color,
Exactly.
right? Versus, yeah, if you read a lot, if you're highly educated and English is your only language, then you get a huge effect. Yeah, so the Stroop task is great, but in practice and research, I think the Simon task, there's other tasks like the flankers task, is just a little bit cleaner.
Yeah. Yeah, so the core statistical procedure in this paper revolved around the deconstruction of midfrontal theta into individual components. And to do this, you use this method known as GED, which stands for generalized eigendecomposition. Could you explain, generally speaking, what signal decomposition or source separation is, what it's used for, and then specifically in this case, why you favored GED over other, I'd say, more common dimensionality reduction feature extraction techniques like ICA and PCA, which are also mentioned in the paper?
Yeah, great. So generalized eigendecomposition is it's a really long word, so apologies for that. But the idea is that, so let me go back to neuroscience and electrophysiology for a moment. So these electrical fields do not just project directly from a spot in the brain only to this one electrode that's directly on top. Instead, the electrical fields just spread through volume conduction and tissue conduction. So that means that when you have some neural generator right here, you can actually measure the electrical field and magnetic field at basically everywhere on the head. And then you have multiple generators, so multiple sources in the brain, multiple like equivalent dipoles or circuits in the brain, generating electrical activity simultaneously. Their electrical fields will sum at the level of the scalp. So that means that any one electrode on the head actually measures activity from lots of neural populations all around the head. The signal is strongest for the neurons directly underneath, and it's going to be weaker for neurons that are further away, but you can still measure the brain tissue that's further away. Okay, so now, imagine you have like these overlapping simultaneous patterns in the EEG data, in the topography, in the spread over space. How do you separate these different patterns? The traditional way to do that is through experimental controls. Basically, you have like, you experimentally separate vision from audition, right? So hearing versus seeing. And then in your analyses, in your data analyses, you just subtract the two maps. And whatever is common gets subtracted, whatever is unique is highlighted. So that's fine. That's a great approach. But that won't work for our case because we started to suspect that there are multiple generators, multiple circuits in the brain that are generating midfrontal theta. So they are close to each other spatially and also operating in the same frequency band and happening at the same time in the same kind of, engaged by the same cognitive process. So those kinds of just very simple subtractive methods don't work. Okay, so we have these overlapping spatial patterns. If these spatial patterns really are distinct, they can be similar, they can be correlated, but they're distinct because they're coming from different calculations in the brain. Then there are multivariate techniques that allow us to basically separate these patterns and see that they're similar to each other, they're overlapping, but they actually are distinct. So to do that, yeah, you need basically to push the data into some other kind of space, and then basically find vectors in that space, like basis vectors in that higher dimensional space where the two patterns are actually linearly separable. And that's what generalized eigendecomposition does. It pushes the data into a space where we can identify basis vectors that are unique for these different components, even though in the sensor space, they are mixed. So we can unmix them basically by pushing the data into this other space using some linear algebra techniques like covariance matrices and eigendecomposition. Okay, so now I can say a little bit more about that technique. So the way it works is by comparing two different covariance matrices, and a covariance matrix is all of the pairwise covariances, which are like unscaled correlation coefficients across all of the pairs of electrodes. And the way that GED works is by defining two covariance matrices that you want to compare. And what we used is covariance in the theta band, so around 6 hertz. That's one covariance matrix, and the other covariance matrix is like a reference covariance, and that comes from the rest of the frequency spectrum, so all the other frequencies besides 6 hertz. And
So you called that a broadband, if I'm not mistaken. That
exactly,
was your broadband range.
exactly. So we have a
Right.
theta
Okay.
covariance and a broadband covariance. And the key thing is that these data are recorded simultaneously. It's literally exactly the same data, same sensors, same subjects, same time points, same everything, except for this narrowband filtering in theta. Now, any pattern that is in both of these covariance matrices will be ignored by the generalized eigendecomposition. And what the GED will identify is the patterns that are unique to theta that are not present in any other frequency. And that is how we got these patterns that are unique to theta. Now, ICA and PCA are also fantastic techniques. They rely on different assumptions. They will reveal different patterns in the data. PCA is blind in the sense that you don't start with two covariance matrices. You just have one covariance matrix of the whole data and you put it into PCA. PCA is constrained by orthogonality, so that all the components need to be zero correlated with each other. ICA, there's different ICA algorithms. They all work slightly differently, but mostly what they are looking for is non-Gaussian sources. The assumption of ICA is that true signals are non-Gaussian distributed and mixtures of signals and random noise is Gaussian distributed. So what ICA will do is also a blind technique. What it will do is try to suppress anything that looks Gaussian and boost anything that looks non-Gaussian. But we find empirically that brain oscillations act like noisy oscillators or non-stationary oscillators, and their distributions actually look quite Gaussian. So ICA is actually suppressing the signal that we want to look for, right? Uh, so ICA and PCA are good methods for what they are for some applications, not for this particular application. Furthermore, ICA and PCA are purely descriptive techniques. They are not inferential techniques. So when you do an ICA, you get a bunch of components. You don't get like a p-value or a confidence interval associated with the different components. But generalized eigendecomposition is a hypothesis-driven method. It's used for, or it's amenable to statistical inference. So you can actually get a p-value associated with each component. And you can say, is this significant? Is it meaningful? Is it, sort of inside or outside the noise distribution? And so that was important for us because we want to say not just there are components, it's trivial that there will be, you decompose data, you're going to get components. That's trivial. The question is, are these statistically robust? Are they meaningful? And that level of rigorous statistical inference we get from the GED, and you don't really get it from ICA and PCA. So there's multiple reasons why GED was just the right method to use for this study.
Dude, fantastic explanation. You should be an instructor or something.
Yeah, thanks. I apologize. Yeah, some of the descriptions sometimes get a little bit technical. If you didn't understand, not you, but if some of the listeners didn't really get all the detail about the Gaussianity and stuff like that, then don't worry about
it. Head on over to Sync Express.
Yeah, exactly. Yeah.
No, great stuff, Mike. Okay, maybe let's finish off the methods section. So, like you mentioned, in addition to like deriving these components, it's also important to address different kind of statistical attributes like significance, uniqueness, stability. And you also looked at whether these components were kind of hierarchically organized. Let's maybe finish this section off by discussing how you carried out some of these complementary validation analyses.
Yeah, great. So in this paper, we wanted to argue that 20 years of research is wrong.
Here.
And that's a little polemic. So we weren't arguing that all the research was wrong, but we really wanted to convince people, ourselves and also other people in the field, that the idea that response conflict monitoring is one cognitive neural operation, that it's a unitary thing. We convinced ourselves that that was wrong, and we wanted to show other people that that was wrong. That doesn't invalidate the previous decades of research. It just adds a nuance. Okay, so to do that, we felt that we needed like really, really rock solid evidence, right? And so, so we had a lot of rigorous, possibly even like gratuitously rigorous statistical controls and thresholding and selection in the study to make sure that we were being really careful and really selective. So that's why we filtered components according to, if the eigenvalues were too close to each other, then we threw out those components. If they were not statistically significant at p less than 0.05, correcting for multiple comparisons across 300 plus sensors, then we threw out those components. If they were not midfrontal, then we threw out those components. So if they were not sufficiently theta modulated, then we threw out those components. So yeah, we did a lot of, we basically threw out a lot of components that I think are real in the brain. They just weren't quite strong enough statistically for us to keep them with this really strong threshold. And still, even after all of these multiple stages of statistical thresholding, we still ended up with, like between 3 and 11 components within each individual participant. So that was quite robust. You also mentioned the Granger causality and this idea of like looking at networks. So some of the analyses we had planned before we started the research, some of the analyses we did kind of like post hoc. So we looked at the data and we saw, wow, there's this pattern. And what does it mean? We need to investigate this a little bit more. So we started doing even more analyses than we initially planned on. After finding that each person has multiple components, then we started wondering, what do they mean? How do they interact with each other? Are they synchronous? Are they sort of passing information back and forth? One of the ideas that we tested was that there is one dominant midfrontal, like one CEO of the midfrontal theta network who is kind of guiding and regulating all of these other like subordinate components. So that's where some of the analyses also came from, looking at Granger mass influx and outflux.
Okay, let's move on to the results section. So you kind of touched upon this a little bit. How many components did you guys find on average, and was that number variable across individuals?
Yeah. So I should have said this in the very beginning, but kudos also to Marit Zurwe, who is the lead author on this paper. She is a PhD student in my group. Yeah, she did a lot of the code for this. So she did a fantastic job on this paper. Yeah, so we found, so it's tough to say with, with how do I get it? Like, if you want to know, how do I fly from, I don't know, Berlin to New York? There are planes that fly that route, and there are planes that don't fly that route. So there's a definite number, and it's a discrete number, right? When you're talking about something that's continuous and also noisy and variable, like how many components are there in the brain? It's hard to come up with an exact number. So in the paper, we found in different participants and different people, we found between 3 and I think 11 was the highest number that we found. But that number, I think the true number is much higher because we did so much conservative statistical thresholding. So, the number that we found is 3 to 11. That's the range. But I think the number is probably more like 10 to 20 in general, just based on, like I have intuitions for what I believe when I look at data that, are not necessarily the same as what comes out in the statistical thresholding. I'm sure you have this experience too, right? Like you look at your fMRI maps and you're like, yeah, I believe this because yeah, you have years of intuition that you develop. But then, at the end of the day, you can't just write a paper saying, I think this because I have this gooey, good feeling about, So you have to do some rigorous statistical threshold. Yeah. So, I mean, the answer to your question is 3 to 11 components, and it varied across different people. Why was it so variable? I don't know. It might be that some people really have more components than other people for whatever reason. But I think it's also just in some people we had better signal measurements, right? The signals were just a little higher. Maybe someone's brain skull is a little bit thinner, so the signal quality is a little bit better. So therefore, we had slightly better statistical evidence. So that bumped the components from 5 to 8 or something like that. Yeah. So I'm reluctant to give a definite answer to your question. It's more than one. I will say that.
Midfrontal theta obviously is believed to be involved in processing of conflict, but was there any evidence of that in this work?
Yeah. So we selected these midfrontal theta components, and then afterwards we looked to see how they were modulated by the task and how they were modulated by conflict. And basically, they almost all were. I forget the exact numbers, but most of these components that we identified were significantly modulated by response conflict. So they're more engaged when there were these trials in the task where you're likely to make a mistake. Even though, so I should also mention in this, in the data, we actually excluded all of the errors, right? So every trial that we analyzed was correct, but some of them had were in these conditions where people were more likely to make a mistake, so the high conflict conditions. Okay, so we found that most of these theta components were modulated by conflict, even though they were all the behavioral response was actually correct. One thing that was interesting that we observed in these data that I've never seen before, because I think, all the previous analyses, all the previous studies we were doing, we were analyzing all these components mixed together and treating them as if they were one thing. When we did this decomposition and we separated them, we actually found that some of these components spin up early and fast, and some of the components are a little bit later and a little bit slower. So that was a pretty interesting phenomenon.
Okay, in terms of hierarchy and interaction between the different components, so what was the result of the grandeur causality analysis?
Yeah, so we found these components, right? And then it was like, okay, now what? Like, what does it mean? And is it the case that these different components are, each component maps onto a unique cognitive subprocess of conflict processing? Or are they all working together? Or like, are they just creating this space, this sort of like computation space for calculations to be operating in? Or is it the case that there's one midfrontal theta circuit that is doing most of the work, like the traditional thought, and these other smaller theta components are just like the little, like, slave workers? So that's I briefly mentioned that before. So with the Granger causality, we tried to test that latter hypothesis. So there, the thinking was, we get, let's say we get 5 components. Let's say one component is doing most of the work and the others are just like, the janitors cleaning up afterwards. If that is the case, then what we expect is, this one component is the loudest. He's screaming at everyone else, and everyone else is like, yeah, okay, we'll do the work, right? So, so that was like our idea. What we thought to test that idea was look at Granger causality, which is a way of looking at causal flow of interactions across different circuits. So what we were so our hypothesis is that if there really is one CEO, then there should be one component per subject, per participant, where it's most active and it has the strongest like causal outflow, right? But we actually didn't find that. So we found a fairly even balance of causal inflow and causal outflow. So it seems like not so much that there's one CEO, but more like it's a collaborative board effort. I shouldn't push this analogy too far, but it seemed more like these different components are roughly on equal standing, and roughly equally engaged and similarly kind of talking to each other and transmitting information to each other. So that was an interesting analysis that also changed our interpretation of the results, which we discuss at the end of the paper.
Okay, so let's get to that. So at the end of the day, what did we learn from this paper, and what's left to explore?
So what's left to explore is everything. What we learn, so the traditional idea of response conflict is that it's unidimensional. So you have no response conflict, or a little bit of response conflict, or a lot of response conflict. You can think about it as like just a line, right? And there's this ball that goes up and down the line. There's no conflict, there's a little bit of conflict, there's a lot of conflict. And now imagine, so just think about how little information that stores. You just get magnitude, right? But now imagine you go to a 2-dimensional space. So now you have, it's not just going up and down, it can go, you can be anywhere in this Cartesian plane. You can represent so much more information. And now go from 2 dimensions to 3 dimensions, right? So now you have this 3-dimensional world where you can have this like conflict particle just sort of traveling around. And maybe the further away from the origin it is, the more total conflict magnitude the person is experiencing. But otherwise, there's different pockets of this space where different kinds of computations or calculations or contexts can be incorporated. So basically, this is what we thought at the end of the paper, that you have this space of midfrontal theta, and it's multidimensional, so 5-dimensional, 10-dimensional, whatever. And inside this space, there is no any one privileged axis. So there's no one line that conflict is constrained to. Instead, response conflict is being calculated in this very high dimensional space. And that allows for just a huge amount of information and context and planning and so on, like reward, emotional modulation, so on, to be integrated into this conflict calculation. So that is how we concluded the paper, and that's kind of the update of our thinking. Yeah, what's left to do? I mean, what are these different dimensions? What is being calculated inside this space? Yeah, so we tried to do some analyses. I think we were just partly, you can't do everything in one paper, right? Science progresses slowly and through accumulation. We were also starting to work on animal models where we can actually implant electrodes directly into the brain and measure LFP, which is like the sort of EEG, but inside the brain, and also neural spiking at the same time. We did some of that work, and it was great. Actually, we reproduced the results in the sense of finding multiple components inside the rat medial frontal cortex that were qualitatively quite similar to what we saw in humans. To be honest, we never actually really finished that work because, because eventually I left academia for other reasons, so I didn't continue that research. But I think there's a lot more work that could be done.
Mike, in academic circles, there's this kind of running joke that whenever you submit a scientific paper and then you hear back from the reviewers, usually reviewers 1 and 2 are very chill. It's like, oh, okay, very good job, very good, well documented, well analyzed, all sorts of stuff. Maybe like fix a typo here and there, add like this very superficial kind of layer of analysis here. And then you get to reviewer 3, and this person is the real hard ass who will like really, really wail into your work. So allow me to just put my reviewer 3 cap on for a second. How would you respond to criticism that the sample size might have been too small to draw robust conclusions? So unless you replicate these findings in larger cohorts, at the end of the day, what you found only applies to this small sample of 9 individuals.
There's always one reviewer who has a bad day and thinks he can take it out on the authors. It's certainly a fair criticism. I will say sometimes we don't get to the ideal data acquisition or experimental acquisition for practical, pragmatic reasons in science. Sometimes experiments are really expensive to do. Sometimes they're just practically very difficult. Sometimes we just run out of funding, right? Maybe the PhD student or postdoc who's working on the project, their contract runs out and they want to get on with their life. So in this case, this study was done in collaboration with colleagues of mine at UC San Francisco. So we were traveling from Amsterdam to San Francisco to collect these data. It's really difficult to get simultaneous EEG and MEG. It takes a long time to prepare each individual. It takes a long time to like sit in the doer, the MEG scanner. It's just very different. It's also hard to find people to do this because it's a pretty brutal experiment. You can't just put a cap on someone because the cap doesn't fit inside the MEG. So we had to paste each individual electrode onto the head. So it's just a pretty, uncomfortable and long period of time to do this research, and just not so pragmatic. So basically, for practical reasons, it wasn't really feasible for us to get a sample size of like, I don't know, 30 or 40 or 50 people. That said, when you need large sample sizes, it's because you want to identify patterns in the group that are generalizable to other people. In our case, we were focused on within subjects analyses, not on across subjects analyses. So all the key results were done on analyses within each individual person. So that is so now we had we actually started with 10 people. We had to exclude one for technical reasons. So we had 9 data sets in here. And the findings that we observed were present in 9 out of 9 subjects. So every single person we looked at showed the same pattern. It was slightly different across different subjects, but overall it was basically the same findings. So I think when you are trying to do group level analyses, 9 is usually not enough. But in our case, we were doing within subjects analyses. So I think 9 out of 9 is pretty convincing. But yeah, it's a valid criticism. I mean, would it have been better to spend an extra year and get another 10 subjects? Yeah, that would have been great. But yeah, that runs up cost and time, and my time, Marit's time, the first author, the technicians and my colleagues in San Francisco. So yeah, there are also just practical concerns.
Yeah. Yeah. All right. Let's move on to a few viewer submitted questions. This one is from Stella Stillwell, and I guess I can kind of like paraphrase what she asked. And this is a theme that you've kind of discussed. How can you tell the difference basically between actual conflict specific signal in the brain and noise as far as these like theta components are concerned? So could one component be noise, and could the other one be like a relevant cognitive computation? And then how can you tell the two apart?
Yeah. So thank you, Stella, for that question. It is not trivial to make that distinction. Part of why it's not trivial is just because of like theoretical reasons that what looks like noise might actually be signal. You're just not thinking about it in the right way. And part of it is statistical. So when you do this kind of component decomposition, this generalized eigendecomposition, the number of components that you get from the analysis equals the number of channels that you put into the data. So if you start with 330 sensors, the decomposition is going to have 330 components, right? And so a lot of those are noise. And I said we found, let's say 5, 3 to 11, I'll just say 5. We found 5 components per subject. It's not that 5 components came out of the result or the analysis. 330 result components came out. It was just that 5 of them, which is like 2%, were actually meaningful. So how do we know? This is something that we struggle with, that I struggle with, because also, as I mentioned a bit earlier, there were more components in the data that I personally believed were real, but we excluded them from and real in the sense of really reflecting some neurocognitive function. But we excluded them from the analysis because we didn't have enough statistical support for them. So yeah, part of the separating signal from noise comes from doing p-value thresholding. Part of it comes from selecting based on component topography, so the spatial layout of the topographies. Part of it comes from, we did a split half analysis where we took each component and we split the data into 2. We took the even trials and we took the odd trials. And then, or maybe the first half, I forget exactly some of the details, but basically we split the data set into 2. And then we ran the same analysis twice. So now we have 3 sets of components from the full data set and from 2 halves of the data set. And then we see, is there actually a match? Now, if these components were just noise, then we would expect the three sets of components to look different from each other because it's just noise. We're just cutting up noise in different ways. And there what we found was, I think it was close to 90% of the components that we identified were observed in the full data set and also reproduced when we split the data into two halves. So that's pretty compelling that these are not just noise. But that said, I mean, to be honest, we could have used different statistical thresholding techniques and we might have come up with some slightly different results.
Yeah. All right. Maybe another one. This one is from not reviewer, viewer, Yadgar, and he asks if you had the opportunity to, or the possibility to scan anyone, living or dead, who would it be, and what would you expect to find?
Yeah. So thanks also for that question. Interesting. So there's two approaches that we take to understanding the brain and understanding the mind in psychology and in neuroscience. One approach is we say, individuals are unique, every person is special, but we want to discover the common patterns that are present in everyone. And therefore, that's also why you want to get a big sample size so you can generalize. So therefore, you don't want to measure any one person. You want to measure every person, right? To see what is common across people. That's one approach. Another approach would be to take extreme cases and use that as a way to highlight what the brain is capable of and how these extreme cases teach us something about normal brain function. And here you can think about like studying geniuses to see, like a chess grandmaster, to say like, how does that brain work and how is it the same as, chess novices and how is it different from chess novices? And then you learn a lot from these interesting case studies. So if I had to, like one person, I would prefer to be able to analyze 1,000 data sets than just one data set. But I guess one person, maybe like a psychopath someone who just doesn't experience a lot of response conflict. Like it's a very normal thing. We say you make a mistake and you feel bad, even if it's like a typo when you're writing an email or something like that. There's still something in your brain. There's a little voice that most of us have in our heads that says, I fucked up, and some people, maybe it's worse, right? Some people might think, I'm stupid, I can't do this, I'm terrible, right? But a lot of us think like, yeah, okay, it's just a little typo, whatever. But I think there's people who just, they don't have these kinds of concerns. They don't have these kinds of thoughts. And so, maybe someone like that would be interesting to study as a case study of where this system seems to be unengaged, or maybe the system doesn't exist, right? So when a psychopath makes a mistake, do they still have midfrontal theta? But it's a signal that's generated in the brain, but other systems in the brain are not using that signal. The signal's being ignored. Or maybe they just don't even have that signal in the first place, so they don't recognize that they're making mistakes. So maybe I should say, there is a part of research on midfrontal theta that is focused on different patient groups, like with ADHD, with autism, with schizophrenia. So it is possible that the kind of research I'm describing has been done, and I'm just not aware of it. So if that's the case, then my apologies to those scientists for me not citing their work.
Well, now you got me thinking that one person whose data were dropped, like, could they have been a psychopath? Like they had corrupt data for a reason?
Well, they were all people who were affiliated with UC San Francisco, so they could all be psychopaths.
All right.
Oops, did I say that out loud? I'm just No,
Yeah.
they're all very normal, fantastic people.
Of course. Mike, so that's it for the paper. Amazing. Now, tell me about, let's finish off with this. Like, what are you doing now? I know you have a book coming up on large language models. You're busy with Sync Express. What's happening? What's good?
Yeah, so I mean, there's a much longer story behind this, but basically, over the course of years, maybe like 5 to 8 years or something, I kind of lost my passion for research and gained a passion for teaching. And I wanted to do teaching that was more um, global, more broadly accessible than just teaching to, very lucky, privileged students at a, particular university in the Netherlands. So that and a bunch of other factors, I ended up leaving academia and basically just focusing on my company, which is called Sync Express. Yeah, so I've been making online courses and writing books, and also in the past 6 months, writing shorter posts on Substack, which is how you and I met. So yeah, then I have a course. It's a really long but very detailed deep dive into LLM architecture and training and mechanisms on Udemy. And I wanted to write a book that is not the same as the course, but kind of similar. The idea of the book is that, if you want to learn how large language models work, most of the existing books and courses basically take one of two approaches. One, they show you how to build a model from scratch and train it from like Wikipedia data. And two, so that's a lot of, it's a lot of detail about architecture, about PyTorch library in Python, and about gradient descent and so on. That's one approach, building a model from scratch and training. A second approach is to focus on integrating LLMs into applications. And that's basically just, setting up APIs to have your phone app talk to ChatGPT behind the scenes. And those are fine. Both of those approaches are great. But I think there is a gap in the education space for learning about LLMs. And that gap is for people who have some background with machine learning or data science or statistics. They don't have a background in like gradient descent, right, or PyTorch. And maybe they're not interested in training models from scratch. They want to start with existing models. They're good with data, right? A lot of people have a background working with data, machine learning, and so on. So I want to help people with that kind of background learn about LLM. So the idea of the book that I'm just finishing up now is that we take open source LLMs. So these are like older models, right? Because the frontier models are not, they're proprietary, so they're sort of hidden away. But the open source models, you can access all the weights, and you can prompt the models the way you would prompt ChatGPT. And you can measure all the activations from every like neuron or every calculation inside these LLMs. And you can bring all of those data into Python, and then you can apply machine learning techniques, regression, clustering, t-tests, dimension reduction, all sorts of things you can do with these data. And that helps people understand how these LLMs are put together, how they process text, how the transformer works, the attention mechanism, and all this fun stuff. So yeah, that's the idea of the book. Yeah.
Wonderful. Wish you all the best on this endeavor, and hopefully when the book is out, we can do this again.
Thanks, Will. Yeah, and thanks again for inviting me. I had a good time here.
Yeah, it's been great, Mike. Do you have any final words before we say goodbye?
Uh, no, keep listening to this podcast. It's good.
Good stuff. All right, Mike, have a good one.
Thanks, bro. You too.